Hello, Assalamu alaikum. In this video, we will understand about the digestion of starch. It is known as the carbohydrate. Starch is carbohydrate made up of amylose and amylopectin, the long chain of the glucose, bind with a glycosidic bond after the condensation reaction in the plant. And in this way, these provide the substantial energy 4 kilocalorie per gram. And 45 to 60% of dietary carbohydrates is in the form of starch, which is polysaccharide. So the poly mean many, saccharone mean sugar. The polysaccharide mean the long chain of the sugar, many sugars. And the molecular weight of this polysaccharide is more as compared to the simple sugar. This is the starch containing grains, contains starch and fibers. Starch, which basically is the amylopectin as a branching, while the amylose is the linear chains of the glucose. The starch is contain amylose and amylopectin, and the chain of glucose. Also, the short chain is known as the dextrin. Dextrin contain alpha 1 to 6 linkage also. So remember this, whenever the starch will be entered into your oral cavity, digest partially, and the sublingual gland, parotid gland, which that produce the sodium bicarbonate as well as amylase and also saliva, which that will lubricate and create the basic medium, will lead to when food is chewed, it is mixed with saliva, digestive enzyme like ptyalin, alpha amylase, 5% digest in this oral cavity. After this 5% digestion, the partially digest this food will be lubricated to become a bolus and easily enter into the esophagus for swallowing because the friction will be decreased. So the mastication is very important for the injury of esophagus prevention. The starch into disaccharide maltose and other small polymers of glucose that contain their 3 to 9 glucose molecule. This 9 to glucose molecule, the monosaccharide also you can use the glucose, galactose and fructose. The formula of the glucose, galactose and fructose is here. Stop the video to watch the difference of that. Disaccharide is can be a sucrose, lactose and maltose. Di mean 2, saccharose mean sugar, while the mono mean 1, saccharone mean sugar. The glucose and fructose combine to sucrose. Lactose is the galactose and glucose glycosidic bond. Maltose is the glucose to glucose linkage. Polysaccharides can be a starch, is amylose and amylopectin. Whenever they go into the esophagus to the stomach, the pH will be drop. For this enzyme activity, it is impossible. So the stomach contain acidic medium while the oral cavity contain a basic medium. The activity of salivary amylase is then stopped by acid of gastric secretion because the amylase is essential inactive as an enzyme. Once the pH of as an enzyme falls below about 4 will lead to inhibit the digestion, then the go chyme after the bolus to chyme conversion go into the duodenum after the protein digestion, but the glucose will not digest completely, but duodenum will work for digestion further through pancreas secrete the alpha amylase. Amylose also is produced, and in this way, remember, let's begin to understand the lumen of the digestive tract, the GIT lumen. This lumen contain enterocytes. These enterocytes in the form of the villi. This villi is the smaller portion of the villi I am drawing here, and which that contain microvilli. It is known as the brush border. That is why it is known as microvilli, and it is also known as the brush border cell. These brush border cells contain a brush border enzyme for further digestion of the starch, the further digestion of the glucose like disaccharide and trisaccharide. The brush border, the microvilli here, and this is the lumen. Let's understand about the further explanation of the amylose, which does contain alpha 1 to 4 link, cannot be cut by amylase. So the amylase will not cut, but alpha amylase will work and it secrete from the pancreas. Anyhow, This is the capillary and this is the lacteal, which that are present in the lamina propria of the digestive system, and it is basically used for the absorption of glucose, and the lacteal is used for the fat absorption, but in this video we will not understand about the fat. This is the interstitial space between the blood circulatory system and the enterocyte. This is the mucous membrane which that protect the pathogenicity, a barrier immunity. And in this way, the basically you can say the bacteria will not invade into the enterocyte to cause infection because the barrier immunity. This is a different type of the starch containing product, which that is alpha 1 to 4 link cannot be cut by amylase. So in this way, this is the amylose, and the alpha amylase is used for the cutting. And in this way, after this digestion of the starch, exocrine hormones secrete into the duodenum from the pancreas, and in this way the amylose, partially digested amylose, will convert into the maltotriose and maltose. Maltose is the two glucose containing basically glucose containing residue, while the maltotriose is three. While this is the lactose containing lactose from the milk, which that when you will consume milk contain glucose and galactose. Galactose is the difference is that the carbon number 4 containing hydroxyl group change. So on the other hand, this is the also alpha amylase digests the amylopectin, but the alpha 1 to 6 linkage will not digest. In this way, this is the terminal alpha 1 to 4 linkage, and alpha 1 to 4 linkage will be cut here, will produce the alpha limit dextrin. This alpha limit dextrin contain a 2 to 3 glucose branching will 2 to 3 glucose residue will combine with the 1 6 linkage, the 1 6 beta linkage. In this way, this is the glucose and galactose. In the presence of lactose, which that is the lactase enzyme are present in the brush border cell, which that will cut and digest to produce glucose and galactose, will be entered through GLUT1 channel and symport GLUT1 channel through ATP used to sodium move inside, and glucose will be entered into the enterocyte. While on the other hand, the maltotriose will be converted by the maltase enzyme, and to cut the simple glucose will move via a same mechanism. But the sodium exchange again back toward the opposite, and the when 2 potassium will be entered for the 3 sodium move inside. 2 sodium move inside, so the 1 potassium is involved for it, while the sucrase enzyme used to digest the sucrose, and in this a GLUT5 channel is involved for the fructose influx. It is facilitate diffusion. After this, enterocyte contain a more sodium, but the sodium is not intracellular cation, so that is why the potassium exchange to move out, but inside the when move the glucose into the blood circulatory system through GLUT2 channel. In this way, the sodium potassium exchange will occur. The 2 potassium will be moving to the enterocyte from the blood circulatory system, while the 3 sodium move into the blood circulatory system. And in this way, extracellular cation and intracellular cation will be homeostasis and exchange. This is the interstitial space. Remember, the interstitial space is involved also for the exchange of potassium and sodium. But the GLUT2 channel is involved for the absorption of the glucose and fructose and galactose into the blood circulatory system, while this is a capillary and this is the endocrine cell. The goblet cell produces mucous membrane, while the enteroendocrine cell produces the GIP. This GIP will be targeted to the pancreas to release insulin before the absorption of glucose. In this way, before the absorption of glucose, the insulin will produce for the ready of the absorption of the glucose from blood circulatory system into the cell. So diabetic person will lead to insulin not produced. So in this way, well to cause produce not more insulin to cause hyperglycemia and lead to cause many complications. While the lactose intolerance is some people genetically lactose intolerance due to lactase deficiency or microbiome unavailability. Anyhow, let's understand about what is the microbiome and how to digest the resistant starch like fibers. Fibers contain a cellulose linkage, so the cellulase enzyme are not present in our body. So how to digest? This is a large intestine contains cecum, contain microbiome, ascending toward transfers to descending to sigmoid and rectum to anus. This is some resistant starch link cellulose short chain and conjugated glucose also not digested by small intestine. So what will be happen? So our our own enzymes, so it can be fermented by bacteria lactobacillus, and in our digestive system our microbiome convert into short chain fatty acids and lactic acids as well as vitamin K and vitamin B complexes. It is the conversion from the bacterial fermentation. From the bacteria the microbiome will convert to healthy and helpful molecule which that will be absorbed easily. So that is why the microbiome also play important role for the normal function of our physiology. Anyhow, in this video, we have discussed about the digestion of starch, the carbohydrates. Carbohydrates contain a glucose residue, the glucose molecule. And this glucose molecule is in the form of short chain or long chain, the polysaccharide, amylopectin, amylose or dextrin. So I hope you make sense about this and see in the next video. For this time, bye. And you can learn a lot of more videos about the carbohydrate. I have a video, I will give link in the description below, and you can learn about the carbohydrate biochemistry. But this video about the digestion of starch, how to starch digest. So thanks for watching. Please make sure to subscribe, like and share. Bye.